Hi everyone! Today's video is about iron homeostasis. Iron homeostasis is the tightly regulated process by which the human body maintains adequate levels of iron. This process helps to meet physiological needs while preventing toxicity from excess accumulation. Iron is essential for so many things in our body, especially oxygen transport, cellular respiration, DNA synthesis, and immune function. However, free iron can catalyze the formation of reactive oxygen species that can damage tissue. Therefore, iron homeostasis is extremely important. Because as humans lack an active mechanism to excrete large amounts of iron from our body, balance is achieved primarily through controlling intestinal absorption, recycling of iron from damaged red blood cells, and regulating storage and distribution amongst tissue. This coordinated system involves the gastrointestinal tract, that is the GI tract, liver, bone marrow, spleen, macrophages, and a network of transport and regulatory proteins. Let's start where iron enters the body, through our diet. Dietary iron enters the body in two major forms: heme iron derived from hemoglobin and myoglobin in animal products, and non-heme iron, found mainly in plant-based foods and iron-fortified products. Heme iron is absorbed more efficiently through transporters such as heme carrier protein 1. This is found in the duodenum. After heme iron enters, iron is released from the porphyrin ring inside the enterocyte. The enterocyte is the word that we use to call a cell in the GI tract. Non-heme iron is typically available in the ferric state, that is Fe3+. Therefore, it must be first reduced to the ferrous state, which is Fe2+. This is done by the brush border enzymes. Specifically, it's called brush border ferrireductases because we want to reduce Fe3+ to Fe2+. Such enzymes are called duodenyl cytochrome B. Once the reduction has taken place, they're taken up by transporters. Specifically, for this case, it's taken up by a transporter named divalent metal transporter 1. For short, it's called DMT1. Within intestinal epithelial cells, iron can be temporarily stored bound to ferritin, or it can be exported across the basolateral membrane into the circulation. Let's talk about how this happens, whether it be exporting iron into the circulation from enterocytes or from other macrophages or hepatocytes that store iron. This is mediated by a transmembrane protein called ferroportin. This is actually the only known cellular iron exporter in mammals. Once iron passes through the ferroportin, it is oxidized back into the ferric form, that is Fe3+ form, by ferrooxidases either in the intestines or ceruloplasmin in the plasma. This oxidation allows iron to bind to transferrin. Transferrin is the principal iron transporter protein in blood. Transferrin delivers iron back into tissue by binding to transferrin receptors on cell surfaces. One of the primary receptors are called TFR1. Once transferrin binds to the transferrin receptor, this complex is taken into the cell by endocytosis. Inside this endosome is an acidic environment. The acidic environment promotes iron release from transferrin. The iron molecules are then reduced back to Fe2+ and is transported into the cytosol for metabolic use, while the transferrin and the receptor are recycled back to the cell surface. Most of the body's iron, about 2/3 of it, is incorporated into hemoglobin. Therefore, erythropoiesis, which is the process of making red blood cells in the bone marrow, is the largest consumer of iron. So you may wonder what happens to the iron in damaged or dead red blood cells. These red blood cells are removed from circulation by either macrophages in the spleen, liver, or the bone marrow. This allows efficient recycling of iron from hemoglobin. This recycling pathway supplies far more iron each day than our dietary absorption does, and therefore it underscores the central role of homeostasis with regards to iron. Macrophage-derived iron is either stored as ferritin or hemochromatosis, or is exported via ferroportin to transferrin for reuse in the blood as new red blood cells or for other metabolic processes. Given that iron needs to be very tightly regulated, we also want to look at which molecules are involved in regulating this process. And that's where hepcidin comes in. The liver acts as the master regulator of the systemic iron balance, and this is done through the synthesis of a peptide hormone called hepcidin. Hepcidin controls plasma iron levels by binding to ferroportin. Remember, ferroportin is the protein that's involved in letting iron go out of any type of cell into the circulation. So hepcidin binds to ferroportin and controls it in a way. When hepcidin binds to ferroportin, it induces its internalization and degradation. So it reduces iron export from enterocytes, macrophages, hepatocytes, or any other cell that's containing the iron molecules. When body iron stores are high, or during inflammation or during infection, hepcidin production is increased. When hepcidin is increased, this leads to the decrease of dietary iron absorption and sequestration of iron within storage sites. So in a nutshell, when serum iron's high or during inflammation or infection, more hepcidin is produced, and with more hepcidin, ferroportin is blocked. Conversely, when there's iron deficiency or hypoxia or increased erythropoietic demand, hepcidin production is suppressed. So hepcidin production is low, and ferroportins are able to act in its way and let go of the iron that's stored inside cells. This allows for more iron to enter the circulation to support red blood cell formation. At the cellular level, iron metabolism is also regulated by a system that's called IRE and IRP system. This system fine-tunes the expression of key proteins. Proteins like ferritin, transferrin receptor, and DMT1 in response to intracellular iron availability. When iron is scarce, which is when iron is low, the IRPs bind to the IRE's messenger RNAs. So this means that the iron regulatory proteins bind to the iron responsive elements on messenger RNAs, and this is to increase the iron uptake and decrease iron storage. When iron is abundant, so iron levels are high, iron in itself prevents this binding. So it favors iron storage and it limits further acquisition of iron. This post-transcriptional control ensures that individual cells maintain sufficient iron for essential processes without accumulating harmful excess. Disruption of iron homeostasis can lead to clinically significant disorders. Iron deficiency is often caused by inadequate intake, malabsorption, or chronic blood loss results in depletion of iron stores and impaired hemoglobin synthesis. This ends up leading to microcytic anemia and reduced oxygen carrying capacity within red blood cells. In contrast, excessive iron accumulation occurs in conditions such as hereditary hemochromatosis, typically due to mutations affecting hepcidin signaling or ferroportin regulation, or even in a person that receives a lot of blood transfusions. This ends up leading to progressive iron deposition in organs like liver, heart, and pancreas. This can cause cirrhosis, cardiomyopathy, diabetes, and other complications due to oxidative tissue damage. Overall, iron homeostasis represents a complex multi-organ system. It integrates dietary absorption, plasma transport, cellular uptake, recycling, storage, and even hormone regulation. Through the coordination of actions of transporters, storage proteins, regulatory hormones, and intracellular sensing mechanisms, the body maintains iron levels within a narrow physiologic range. This balance is extremely critical for sustaining oxygen delivery and metabolic activities while protecting tissue from potentially toxic effects of excess free iron. And that's it for this video. I hope it was helpful for you. If it was, please make sure to like and subscribe. I'll see you in the next video. Bye!